Students, welcome back to another biochemistry classes dealing with the molecular techniques. Today we will go on to another molecular technique, that is PCR or polymerase chain reaction. So this polymerase chain reaction, you will have to write it down in your record notebooks. Write down the theory that is taught in the rule side, and the diagram please draw it on the blank side, right? So when we talk about polymerase chain reactions, it can also be called as PCR. So PCR was first developed by Karry Mullis in 1984, and it is considered as a basic tool for molecular biology. So here the laboratory technique generates large quantities of specialized DNA from a target DNA sequence. So this is a cell-free amplification technique for synthesis of multiple identical copies of DNA which is of interest. So next we'll go on to what is the principle of PCR. The DNA of interest is first denatured and the two DNA strands are separated. So this process we call it as denaturation. Now after this, the two strands then are hybridized with a primer. So primers are nothing but around 20 to 30 nucleotide bases, okay, which gets attached to the denatured DNA strand, and this process we call it as renaturation. Now what happens after this is the process of multiplication of the DNA strands. So the primer attaches to the template duplex and leads to the synthesis of DNA, and this process is synthesis of the DNA or amplification of the DNA. Now after these three steps, this is again repeated number of times, that is, and from the DNA target, and you can get millions of the target DNA which is amplified. So the main three processes of in PCR is denaturation, renaturation and synthesis. Now what are the requirements of PCR? We have a number of essential requirements for PCR, that is the target DNA of 100 to 35,000 pp in length, and then you require primers, right? So primers are sufficiently accurate. So if they are not sufficiently accurate, then the DNA synthesis will not take place in the right way. So the reliability of the PCR depends on the primer that gets attached to the DNA strands. So now what is a primer? It is nothing but made up of 20 to 30 or 70 to 30 nucleotide base pairs, and they are complementary to the regions, that is the target DNA. Then you require the 4 deoxyribonucleotides, that is deoxy-ATP, deoxy-CTP, deoxy-GDP and deoxy-TTP. Then you require the enzyme, that is DNA polymerase enzymes, which can stand at a temperature but doesn't dissociate at any high temperature. So here we are using around 95 degrees centigrade. So these are the 4 requirements of PCR. When we go on to the real procedure, we said that the first one, so the PCR involves repeated cycle for application of the target DNA. So now first the target DNA gets denatured. So we raise the temperature to about 95 degrees for 1 minute, and the two DNA strands gets separated. Now after it gets separated, the next process is renaturation or annealing. Now the temperature of the mixture is slowly decreased to 55 degrees centigrade. Then the primer basis with the complementary region flanking the target DNA is attached, and this process we call it as renaturation or annealing. Now after this, the high concentration of the primer ensures annealing between the DNA strand and the primer rather than the two DNA strands that gets separated. initiation of the DNA synthesis occurs at the 3' hydroxyl end of each primer. Right? So at this, the primer are extended by joining the bases, which is complementary to the DNA strand. And after this, the temperature has to be kept as optimal as the required by the enzyme DNA polymerase enzyme. Now this temperature is around 95. So after we cool it is 55. So the optimum temperature is around 75 degrees for the dermis aquatis DNA polymerase enzyme, whereas for the E. coli DNA polymerase it is around 37 degrees centigrade. Now the reaction can be stopped by raising the temperature for more after 95 degrees centigrade, but this doesn't happen. This synthesis process in the PCR is quite comparable to the DNA replication of the leading strand. Now after this, the new strand is joined to each primer and it is beyond the sequence that is complementary to the second primer. Now these new strands are referred to as the long templates and they will be used as templates for the second cycle of the PCR. Now at the end of the second cycle, the long and the short templates, that is the DNA strands with the primer sequence at one end and the sequence complementary to the other end are formed, and this after the third cycle, the original DNA strands along with the long and the short templates are the starting material for the next cycle of the PCR. Now the technique of denaturation, renaturation and the synthesis are repeated multiple times again and again, and the target DNA is amplified. So this, it is estimated at the end of the 32nd cycle approximately of the PCR, 1,000,000-fold of the target DNA is synthesized, right? So now when it is multiplied and synthesized around the 32nd cycle, 1,000,000-fold of the target DNA is synthesized. Next we'll go on to the procedure of DNA when we look at this flow chart. So this is the original DNA strand, that is 5', 3' end, and on the opposite direction 3' into the 5' end. So here it gets denatured, and because of this there is DNA which is raised at 95 degrees centigrade and both the strands gets separated. Now after the separation, the DNA primers gets attached to the 3' end of both the separated DNA strands, and after that it is cooled to 50 degrees to annealing with the primers. Now subsequently after that there is synthesis of the DNA strand in the opposite direction. You can see from the 5' to the 3' and this side it is from the 5' to the 3' and two new DNA strands are synthesized which are complementary to the target DNA sequence. So this is the process of the PCR technique. So in the next flowchart you can see here the region to be amplified, or we can call it as a target DNA. Okay. Now this target DNA is made up of double strands. So by melting the DNA at 95 degrees, the two DNA strands are separated. Then you add the primers and at 50 degrees centigrade the annealing of the primers takes place. Now after that you add the tag polymerase, that is the deoxynucleotides, and you anneal with that 72 degrees centigrade. Now after that 72 degrees centigrade, the two DNA strands are attached to the primers as you can see here, and then you can see that newly synthesized strands are a little bit longer than the template or the target DNA strand. So after this the DNA strand is melted at 95 degrees and the primer. So this is the old DNA strand. This is the newly synthesized DNA strand. As you can see, it is longer than the complementary strand. So because of this, the newly synthesized DNA strands, which are the long and the short strands, act as a template for the next cycle of PCR technique. So then after this is the first cycle. That is where you get 4 strands. Subsequently, after the second cycle, you get 8 strands of the target DNA strand. Then after the third cycle, around 16 strands are synthesized. So this DNA at 95 degrees added. Then you have the primers and then annealing at 50 degrees centigrade. And then again the primers are added. Deoxynucleotides are added. TAQ, that is the polymerase enzyme, is also added at 72 degrees centigrade. And so now you can see from the target DNA, from the first cycle you have 4 DNA strands that are synthesized. Now after that, the second cycle, you can see around 8 newly synthesized DNA strands, and after the third cycle you can see around 16 strands of the target DNA is synthesized. Okay, so see to that in your records on the unruled side you draw both the diagrams of flow chart that is being shown in this PPT. Okay, and then the whole process of the theory part of whatever is told about the PCR technique, please write it in the rule side, right? So this is the whole process of PCR. So we'll sum up the whole process once more. The 2 DNA strands are denatured and it is separated. So this leads to 2 separate DNA strands, that is around 95 degrees centigrade. And then these 2 separated DNA strands are attached with a primer, that is nothing but 20 to 30 nucleotide bases. Okay, so this primer is attached to the 3' end of both the strands that are separated, and after that there is synthesis of the new DNA strand. So this 2 DNA strands will have long template and a short strands. So this we call it as the next, they form the template for the next cycle of the polymerase chain reactions, right? So this is the whole process by which there are synthesis. After 3 cycles, around 16 strands of the target DNA is synthesized. So around 30-second you get around millions of the target DNA that is required for the test, right? So this is about the whole process of the PCR. Next we go on to, there are certain variations of PCR. Okay, a number of them, variants. So you have the nested PCR, you have the inverse PCR, then you have the anchored PCR, the reverse transcriptase PCR, asymmetric PCR, then you have the real-time quantitative PCR, then you have the random amplified polymeric DNA, that is RAPD, then the amplified fragment length polymerization polymorphism, then you have the rapid amplification of cDNA ends, or we also call it as arrays. So these are the different variants of PCR technique. So we went with the requirements for PCR, then we finished with the procedure of the whole PCR, right? And then the flow chart, then we finished with the variations of PCR. Next we'll go on to the applications or uses of PCR. So PCR technique is used as a diagnosis of bacterial and viral infection. So examples like early phases of tuberculosis, then you have Hepatitis C, Cytomegalovirus, and HIV, where you use in cancer detection and also the follow-up identification of mutation in Oncosuppressor genes such as P35, retinoblastoma and leukemia. So in cancer, it helps in both the diagnosis and also of the follow-up of the patients whenever they come for treatment. PCR is useful. Then for the prenatal diagnosis of inherited disease by amniotic fluid analysis of the chorionic villus sample is being taken. That is the amino synthesis you do. Example, the sickle cell anemia, beta-thalassemia and PKU or phenylketonuria. Then you have, it is used in medical legal cases. How it is used is the sample is taken and the DNA is analyzed from the hair follicle or the blood cells, and then we do the DNA fingerprinting. So thus it is useful in medical legal cases, right? So there are mainly 4 applications of PCR. One is the viral and the bacterial infection, and then you have the cancer detection, then you have the prenatal diagnosis of inherited diseases, then you have the medical legal cases, right? So these are the applications of PCR. So we spoke something about the variants of PCR, right? So you also have to know something which we call it as reverse transcriptase PCR, or we also call it as RT-PCR. So RT-PCR is a modified version of the PCR technique, okay, in which the cDNA produced from an RNA sample using reverse transcriptase is amplified. Now RT-PCR, it is used to measure the quantity of the HIV, that is the RNA virus, and the circulating in the blood, which we call it as the viral load, when monitoring the response to drugs or the status of infection in a patient with HIV infection. So these are the uses of reverse transcriptase PCR. So PCR comes as a short note, and also you have to know the different varieties or different variants of PCR and also the applications of PCR. And please write this procedure in your record notebooks on the root side, the theory part, and the diagram in the unrolled side, right? So this finishes with one of the molecular technique. In the next class, we'll go on to another molecular technique. Thank you. Have a nice day.